Right, so I'm going to give you a brief overview of genomic testing. So we've got plenty of time to hear from our lovely people with lived experiences. So I'll give you some links at the end where you can find more information. Um, so I wanted to use this slide from one of my colleagues, kind of looking back at the sort of technical and biological advances that we've had in the last 70 years or so, and sort of comparing them to the advances in CAR technology. So in 1953 was when Watson and Crick first described the structure of DNA. And I've got a picture there of the cars that we might expect to see in 1953, through now to these sort of super fast cars that we have now. And actually our understanding of biology and genomics has improved massively in those 70 years. And now we're able to do whole genome sequencing, so reading the whole of someone's genome. And actually for some individuals where they're acutely unwell, we can do this really quickly in sort of 2 weeks or so. Next slide, please. So just as an overview, so what is our genome? So that is our genetic code, which is made up of our DNA, and our DNA is the sort of chemical building blocks that make up our genes. And we have more than 20,000 genes in total, which each come in pairs, one from mum and one from dad. And those genes are packed inside chromosomes, and we have 23 pairs of chromosomes. And I've just got a picture here just showing a few of the genes that we might see on chromosome 17, but this is certainly not an exhaustive list. And those chromosomes are packed at the centre of our cells. And so when we're doing genomic testing, we're reading that genetic code that's present in all the cells of our bodies. And I've just put a reminder here that we shouldn't forget that we actually, in our mitochondria, so the battery packs for our cells, they also have their own genome. So it's important not to forget that when we're doing testing. Next slide, please. So this is, so I've got a fun fact here about genome, and this is what our sequence would look like. So it's a string of letters, which is more than 3.2 billion base pairs, or 6.4 billion letters in total. And you can see it's a string of sort of 4 different letters or nucleotides. Next slide, please. So when we're talking about genomics, there's lots of different things we can do with that, and this is always improving and changing as time goes on. And one of the things I like the most about working in genomics is the fact that we see people at lots of different stages in their lives. So we might see them when they're born, when they're children, then as young adults, and when they're having children of their own. And so genomics can be relevant at all different points in someone's lifetime. And so a lot of what we're talking about today is whole genome sequencing, where we're reading that whole genetic code, reading all of that DNA sequence, and trying to see if we can find the cause of someone's rare genetic condition. But this can also be relevant, as we said, prenatally. So if we have a baby who's been found on scan to have anomalies, we might want to do urgent testing in pregnancy. And also we'll hear a little bit more from Emma shortly about the new research project looking at newborn whole genome sequencing. If we know a genetic diagnosis in the family, we can potentially offer carrier testing or predictive testing for that condition. And a field that's really growing, and I think will be really interesting in the coming years, will be pharmacogenomics, where we look at different genetic variants that we would have within our genome that may predict how we would respond to certain drugs. Sorry, I'm not sure what happened there. And then maybe predict whether we might have more severe adverse events. We can look at tumours as well and see if we can identify genetic drivers for that cancer, and then that may influence management. And also thinking about metagenomics, which is thinking about different organisms in a sample. So you might be thinking about different infectious diseases, for example. Next slide, please. So in England, genomic testing and all the genetic testing that is available is laid out in the National Genomic Test Directory. And I've got a QR code here that will take you to that test directory. And this is the page you'll see here. And then if you click on the middle one, you'll get up a large PDF file, which you can navigate through to look at different test indications that might be relevant to your patient. And here I've given an example page looking at the testing for global developmental delay or intellectual disability, called the R29 test. And at the top, you'll see that we talk about when it might be indicated, other tests that might overlap, so you can decide if something else is better than what you're maybe looking at currently, and then also who can request it and what type of test it is at the very bottom in that table. And in this example, it's whole genome sequencing, which we're going to focus on more throughout the talk. And next slide, please. So if you go to the panel app, this is where you can see what genes you're looking at. And there are different types of tests, as we've said. So whole genome sequencing, where you read the whole genetic code, but we do focus that analysis in on panels, so you can see which genes that would include. Or sometimes tests might just be panel based, so you can see those genes. And then occasionally we do have some single gene testing as well. And all the green genes are those that will be included in that test. Next slide, please. So all of this is delivered in England through the NHS Genomic Medicine Service and the laboratory hubs. And you can see England here is split into 7 regions, and we're part of the Central and South region, which is kind of the greeny area. So that covers quite a large area, as you can see, through Birmingham in the West Midlands, Oxford, Southampton, Hampshire and Wessex area. Next slide, please. So as I say, I'm going to focus more on whole genome sequencing for the rest of this. So what do we do? And this will be relevant as well to when Emma talks, that at the moment what we do is we take that whole genetic code, all of those billions of letters, and we chop it up into small chunks to read it, and then we put it back together. And then when we've got our sort of long sequence, we then compare that to what we call the reference. So, and then what we're looking for is, can we find differences from the reference in that patient? That's, we might call those alterations, variants. Next slide, please. And we all have lots of shared sequence, but also millions of different variants, changes, and the scientists, when they're doing that testing, need to filter through those, because the vast majority of these are going to be benign and harmless genetic variation. And so that's why we ideally do this as a trio test. And when I say a trio test, I mean the patient with both parents. So we can use parents as a bit of a filter comparison to try and find the genetic variants that might be most relevant to that patient. Next slide, please. So a genetic condition can be caused by just one single genetic change within that genetic code, as you can see here. So there's an A changed to a T. Next slide, please. So I've just done a little table here to give you a bit of an overview of the types of genetic spelling changes that we might identify in our genetic code. And so at the top here, we've got what we call the wild type, or the reference sequence. So that's what we compare everyone to. And as you can see, these have been put in 3-letter words, and that's because that's how we read our DNA. We read it in 3-letter words to then decide the amino acid that's put into the protein. And so we may have a synonymous change, which is basically where we have a single change, but actually the meaning is the same. So in other words, the amino acid is the same. So here we've got add and, so they obviously mean the same. As we saw in the previous slide, we could have a single genetic change in one of these letters that changes the amino acid that put in, and that one single change can be enough, and that would cause that genetic condition in that individual. And as you can see, ham is changed to hat. We can have what's called an in-frame deletion, so that's where we're deleting sort of the multiple of 3 words. So here we would delete 3 amino acids, but it changes, so we've just deleted one egg basically. So we're still putting the ham in. But here, because we've deleted just one letter, we've changed then the reading frame of that. And so eventually that will end in a stop, which will end that protein there. And that's called a frame shift deletion. Sometimes we can pick up a nonsense variant, which means that actually we end that protein straight away. So exactly where that one single changes, that is where we end the protein. And then we can also have duplications. So we'd be adding 2 eggs there, and an insertion where we've got another bit of genetic material that we've put in. Next slide, please. So when we find these variants, then the lab scientists need to filter through to try and see which one's most relevant to the patient. So they think about phenotype, so the clinical features an individual may have. They think about what the gene is, how that protein functions, and whether that variant's likely to affect how that does function in the body. We also think about the general population. So if we're looking for a rare condition, we're not expecting that variant to be common in the population, in which case it's not likely to be relevant. And as we said, ideally we kind of do parents at the same time. So if we have 2 parents who don't have that condition, we might find that it's a new or de novo genetic change in that individual. Or if one of the parents and other family members do have the same condition, then we might find that it's segregating, so it's present in the parent who's got the same condition and other family members. And so that can help us then score it with, so what we'd be looking for is something that's likely pathogenic or pathogenic, and those which are benign and likely benign being more harmless and not so relevant. And then the VUS being the variant of uncertain significance. Next slide, please. Oh, sorry. Yeah, thanks. So there's roughly 4 types of results that we can get from whole genome sequencing. So one is that we find a confirmed genetic diagnosis. So that's where we find that likely pathogenic genetic change, and we're happy that it explains that patient's clinical problems. And then we can talk to them about what that diagnosis means for them and whether it has any management implications, and what it also means for the future if we're able to give that information. The second type of result is what we call an incidental finding. So what that means is we find a genetic variant that could be relevant to them, but it's not actually the cause, not the reason for why we did the test. So if we had a child with developmental delay and we found a genetic change in a gene that's associated with increased rate of cancer predisposition, then that would be an incidental finding. And we can talk to the family and about what that means and whether it's relevant to the wider family as well. The third type of result is no significant finding. So what that means is on this current test, we haven't been able to find a diagnosis. So it may be that there's other testing that we could think about, or it may be that just our understanding isn't quite there yet and we need to revisit things at a later stage. The final type of result is what I sort of intimated about in the last slide, which was a variant of uncertain significance. So what that means is we find a genetic change that's in a gene that could be relevant to the patient's clinical problems, but we're just not sure if that genetic change is affecting how that protein gene functions in the body. And so in that instance, we can't give a confirmed diagnosis, but there might be other tests we can do to decide if it is relevant to that patient. But again, sometimes it can be a case of coming back and revisiting it at a later date. Next slide, please. So I'm not going to go into this too much. Isabel, you might want to press it another couple of times. But this is a record of discussion form, which just goes through the consenting process with the patient and family. And there's a QR code for that on the side. And you need to complete one of these for every person involved. So if we're doing a trio test, that would be the patient and both parents. We'd need a consent form or record of discussion for. Next slide, please. And on that, we also talk about the National Research Library, and this is something that patients and parents, families can decide to opt into. And essentially what happens is the genetic data and health information is anonymised, put into this secure environment that researchers can go into, and it helps us learn more about genetic conditions. But importantly, researchers can't take information away, so it is a secure environment. Next slide, please. And then finally, we have to complete a test order form detailing patients and family members involved, as well as the clinical symptoms or features that they have. And again, there's a QR code for that below. Next slide, please. So I've got here a couple of links to a couple of videos talking through the consent process and also a little bit more information about the research library. Next slide, please. But finally, and probably what we're going to hear a lot more about from people today, is what are the benefits of genetic testing. So having a confirmed diagnosis can be helpful because it ends the sort of diagnostic odyssey, or that some people can be quite a long time. But it can potentially provide therapeutic options in some cases, and can provide prognostics, so information about the future, but also potentially management plans. But in addition to that, it can be relevant for family, thinking about whether somebody is likely to also have the same genetic condition, and we can offer predictive testing or carrier testing depending on that. Next slide, please. So as I said, that was a bit of a whistle-stop tour through, but here is some links at Genomics Education for where you can get more information, and sessions and G-Notes, which provides some really good clinical scenarios that you can have a look at, and also the Central and South website, as well as another interesting genomic fact. So I will pass over to Emma next to talk through more about research. Thank you.
Thank you so much, Hannah. That was a really, really great overview of sort of the testing process within the UK. So I'm going to now hand over to Emma Douglas. So Emma is a genetics clinical research practitioner, also at Birmingham Women's and Children's. And she is also the PPIE lead at Hannah's pilot study Needed. And she's part of the genetics research and audit collaboration. So she's going to tell us a little bit about genomic research and how that benefits patients. So Emma, do I have you on the call?
Yes. Hi, Isabel. Thank you. Hopefully you can hear me.
Yes.
Okay. Yeah, so thank you for the opportunity to speak to you today. So my name's Emma, and yes, I work at Birmingham Women's and Children's NHS Foundation Trust. Next slide, please. So in the NHS, clinical teams work together to provide direct care for patients, and clinical care teams require an evidence base to know how to deliver the best and most effective care, and this is research. Research is recognised as a key driver for improving patient care and medical practice. The National Institute of Health and Care Research was created in 2006, and it's been providing a unified infrastructure to support high quality research in the NHS. Today, research is considered a fundamental duty in the NHS, as reflected in the Health and Social Care Act in 2012. Next slide, please. Thank you. So, with NHS organisations supporting the conduct of research to improve patient outcomes, several trusts hire staff specifically to offer research to NHS patients. And I'm part of this workforce, which is termed research delivery. The research delivery workforce at BWC includes research nurses, research midwives, research data managers, and clinical research practitioners. And I'm a clinical research practitioner, which is an umbrella term now used for a family of roles in research delivery with a research delivery element where the post holder is not previously been recognised registered to a healthcare profession, but the UK Professional Standards Authority now recognises CRPs as an occupational group in health and social care, and has approved CRPs to the Academy of Healthcare Science Accredited Register, which is in 2020. We specialise in consent, data collection and clinical study protocol activities with an emphasis on the care of research participants. So there are lots of types of research studies. They all contribute to a wider base of evidence to lead to the implementation of evidence-based care. Lots of people might just think that there's clinical trials, like drug trials, but there's all sorts of types of studies. In clinical genetics, we most often see observational studies. So observation is collecting data without intervening in some way. And the UK has long been at the forefront of discovery for human genomics and is recognised for its world-leading genetic research. And I've been privileged to deliver several of these studies, including, next slide please, the 100,000 Genomes Project. So the 100,000 Genomes Project was trialling a new next generation technology, hoping and looking to transform the capacity, speed and cost of genomic sequencing. It ran from 2015 to 2018 and was run by Genomics England. So the project aimed to sequence 100,000 genomes from volunteers, including people with rare disease and cancer. This was to establish the use of whole genome sequencing in the NHS and drive change within NHS services to adopt this technology. Next slide, please. The 100,000 Genomes Project was successfully showed that whole genome sequencing could be applied with the NHS, so it demonstrated the feasibility and diagnostic value. And it's now available as part of routine clinical care in the NHS Genomic Medicine Service that Hannah has just mentioned in her presentation. Next slide, please. NHS, as I say, is part of routine clinical care and is nationally available on the NHS to those patients that are eligible. Quite often those who have a rare condition that has not been diagnosed with our genetic testing, and the conditions available for whole genome sequencing testing list it's ongoing and expanding. Next slide, please. Another study that applies whole genome sequencing that is currently running up until March 2027 is the Generation study. So the Generation study is also run by Genomics England in partnership with NHS England and began being offered to NHS patients in 2024 and is gradually expanding the NHS trusts that are able to offer this study to their patients. It aims to facilitate earlier diagnosis and treatment for childhood onset rare genetic conditions. It's trying to support genomic research and explore long-term implications of these conditions. So parents are invited during pregnancy to consent to the baby's participation, and at or after birth, the cord blood sample is collected and sequenced, and that blood sample is what the study uses to run whole genome sequencing testing. If for some reason that gets missed, they do ask to do a Hill prick from baby within 28 days of baby's delivery, but they arrange that with the parents. So with these being rare conditions that they're looking at, it's thought approximately 1% would receive what is considered a condition suspected, because this is a research tool and not diagnostic. But if a condition is suspected and fed back to the family, there is prompt follow-up for diagnostic testing and care through NHS specialists, because this study runs hybrid with the NHS. And there's another section on this slide, Isabel. So all the conditions that they test for usually appear in the first few years of life, can be improved if found early, and have treatment through the NHS. Currently, they test for just over 200 rare genetic conditions, and there is a list available on their website to look through all the conditions, but they were very carefully chosen to be in this study. Next slide. So the endpoints of the study is to sequence 100,000 genomes of newborns in England, establish an evidence base for what screening these babies means for the future treatment and research for newborns, and provide the data needed to evaluate research study outcomes and inform future policy. The idea of screening the newborns through this study is at the earliest point in life to try and pick up these conditions where there is that early treatment available through the NHS. Thank you. Next slide. So although we've mostly talked about whole genome sequencing, there are lots of technologies that are always developing in genetics and genomics. An example of a new technology that is being used at the moment on a research basis is the nanopore technology, also termed long-read sequencing. So nanopore DNA sequencing is a laboratory technique. So it determines the exact sequence of nucleotides or bases in a DNA molecule. So nanopore DNA sequencing involves the reading of a singular DNA strand as they are threaded through extremely tiny pores, nanopores, embedded within a membrane. As the DNA moves through the pore, it creates signals that can be converted to read each base. This approach offers a low-cost, rapid process for studying long stretches of DNA, and a major advantage of nanopore DNA sequencing is the ability to produce ultra-long reads, so much more than produced from some earlier technologies. And we had an in-house study run from Dr. Titheridge, that's on record today, called Needed, that used this technology in a pilot phase to see if they could help patients with rare disease get some diagnosis for their conditions. And also there is a national study, NHR Bioresource. It's a national research resource and bank, but one of the sub-projects within the Bioresource is also using the nanopore technology. Just in summary, research can offer an evidence base for new technologies in genetics and genomics that hopefully can be applied to help patients within NHS cares. Thank you, that's it.
Thank you so much for that, Emma. It's really great to hear how research is sort of used in this way and get a better understanding of it being sort of a part of patient care, and how key that is. So we're now going to move into hearing from people who have lived experiences of genomic testing themselves, either as patients or patient advocates. So opening up here, we have Cara Duffy, who's going to talk about how to get it right when you are delivering diagnoses to your patients or to their carers, advocates, whoever it may be. Cara has her experiences of genetic and genomic testing with her daughter Alana, and she will talk extensively about that. And she's also doing I think it's a PhD, Cara, correct me if I'm wrong, in genomics.
Not quite PhD. Masters I'm doing.
A master's in genomics at the University of Birmingham. So Cara, I'll hand over to you. Just let me know as you want me to click through. And just to note as well, everybody, we will be sharing these slides alongside the recording after the event has taken place. Over to you, Cara.
Okay, so hopefully you can all hear me. Thank you for the introduction, Isabel. So yeah, my name's Cara, and I am a full-time carer for my daughter Ilana, who's nearly 6, so she'll be 6 in September. And she has a rare condition called MAND syndrome. I think the prevalence of that is like 1 in a million or something. So it is one of the rarer ones. Essentially, she's missing one protein, and the chromosome, on chromosome 2, on the Q1 chromosome 2. Yes, I am got a bit of background. My undergrad was in forensic science. Started lecturing forensic science and then genetic profiling process, and then I ended up with a child with a genetic disorder. So I ended up wanting to go now into hopefully genetic counselling. So I am doing my master's in genomic medicine, and can use my own experience for that. Before I do get started, I do want to actually acknowledge all the amazing clinicians that are in Alana's care. They are almost like, not quite extended family, but they are really important to us. And I think it's getting that good rapport with your patients where possible is essential, really. I know that can be difficult, especially with time constraints and clinic appointment times, but it does really help if you can get that like sort of rapport going and a good working relationship. Next slide, please. Okay. So yeah, a little bit of history. So I'll go through our diagnostic history of Alana. She was born at 36 and 6 weeks by a planned C-section. It was advised that she shouldn't go past 37 weeks because I couldn't really feel her move much during the pregnancy. And She was nearly born at actually 28 weeks. Then they were prepping me for a C-section because she had a low STV rate. So that did improve, but as I say, it was decided don't go past 37 weeks due to like that down to heart rate issues. So when she was delivered, I did have the steroid injections because she was like a day of being considered term. She did have difficult breathing and she was sent straight to NICU. And she was there for 12 days in total. 48 hours she was on the high flow oxygen, and then the rest because she was just finding it difficult to feed. We were discharged with her. She did have an NG tube to top of her feeds, but that was removed 2 weeks after discharge. So she would have been about a month old then. One of the first sort of signs for me, for Alana, is she slept all the time. Now, she's my second child, so it was a completely different experience from the start. I couldn't hear her cry. It was barely audible. And I remember being in the NICU saying to one of the nurses, how am I meant to wake up to feed her because I can't hear her? And they were going, oh, well, you'll regret saying that. No, obviously, if she was healthy, I might have, but I would have loved to hear her like proper full belt cry. I had to, for the first few months, I had to set alarm every 3 hours to feed her, just because, you know, she would not have woken up. She wouldn't have had enough feed. Again, during feeding, I did breastfeed, but I couldn't feel her suckle. But I was convinced by the nurses that, well, we can see you swallow. Why can't you feel that? So you just agree. You just kind of go, oh, okay, because it makes you feel inadequate. It's hard being a parent, especially in that newborn kind of blur that happens and lack of sleep and everything else. I did fully breastfeed her older brother, so I was thinking, I don't remember like feeling. I could feel him latch, you know, I could feel him suckle, and I was just like thinking, why couldn't I feel it with her? So, but I was just convinced that she was feeding okay, and you do take at the start. You do take the professional's opinion. You know that they are the experts. So you're just saying, oh, okay, it must be me then. There's something wrong with me. The only way I could feed her though was with nipple shields. So I ended up going to an amazing breastfeeding midwife. Her name's Jackie Scott, absolutely the best breastfeeding specialist known. Because I just wasn't happy with the way she was feeding. I wasn't happy with the shield, and I was just concerned that she wasn't gaining any weight. I mean, she was discharged from NICU, only gaining 10 grams of weight a day. So, you know, she was really struggling to feed and to gain that weight. Can you next slide, please? So that's in the October. I've got just in a little few pictures, like just kind of our little timeline. So around 6 weeks old, I was telling the health visitor and the GP at the checkup, I didn't think Alana could see. There's blindness in my family anyway, so it's not like it was something that, you know, it's still devastating, but like I thought, oh, okay, maybe she has inherited that. Because she wouldn't track lights, but they kept fobbing me off. They kept telling me like, oh, you can see her looking at you, don't worry. I can see her watching you move across the room, but her eyes were moving to the sound, not to she weren't tracking anything. And I was just like thinking, okay. The checkup that she had at the sort of the 6-week checkup, she pretty much failed because like she was really floppy and everything else. But again, there was no concerns at this point from the professionals, but something was niggling at me that something wasn't quite right. So then we move into November. So if you can go on to the next slide, please. So yeah, so when she got to around 10 weeks old, again, I was at that breastfeeding clinic with Jackie, and she asked if she could weigh Alana and picked her up, and she just shared this look with one of her colleagues, and I was like, thank you, almost, because it validated that I knew something was wrong. And I was thinking, what could be wrong? I know that look. I had my own doubts. So then Jackie got on the case to the paediatrician who did deliver her to review her. Unfortunately, he ignored the situation. He said that It doesn't need to review, she doesn't need to follow up. So we then move into December. You can do next slide so you can see where she's at in December. So she spoke to a different paediatrician, and he saw her the next day in clinic. It's her main paediatrician now. He is amazing. He's out of what he does. It was a very brief meeting, that first one, but he saw her in clinic the following week. There were lots of questions, and the tests were ordered an MRI, things like that. There was lots of questions about consanguinity, your family history of illnesses, etc. They were all asked. I'll be honest, even though my background is like sort of forensic science with that sort of, and with the genetic link, I still didn't think there was a genetic disorder. And it's like, I knew something was wrong, but I didn't think a genetic disorder would be wrong, apart from the blindness. Given she was nearly born at 28 weeks, I thought maybe there was a cerebral palsy issue. But you know, that's hindsight, isn't it? I thought I was there mainly because she wasn't gaining weight and it was feeding. I remember thinking, again, there's Crohn's in the family, celiac in the family. So I was thinking, oh, is that why she's not feeding? Is that why she's gaining weight? Lots of things going on, but I didn't think it was genetic at all. Alana was sent for blood today. She had an ECG, she had a heart murmur, and then she was also sent for an urgent MRI was done. That was December in 2019. I did pick up, like, the doctor was saying, like, she said that she got the one dysmorphic feature. She had like a she had nares, inverted nares. But again, I shrugged it off because I've got a little turned up nose. I was thinking, okay, right, yeah, all right, fair enough. But she was very floppy. Again, I put that down for potential cerebral palsy. I wasn't quite sure what was going on. So then we moved into the January. So if we can go on to the next slide. All right, okay. So this is her at 4 months old. You can see she's smiling there. So she, you know, she was becoming a lot more awake. She'd started rolling over as well. So that was like really like quite nice, reassuring. So she was like starting to do some things. But that's when we got the diagnosis of being visually impaired, but they needed to have more scans because they didn't know why she was blind. They did confirm that she was blind, but they didn't know whether it was what part of the anatomy wasn't working. It seemed her eyes were okay, but she just wasn't interpreting the signals. So again, that's pointing to something cerebral going on. Not much else happened really in the January. She was sort of waking up more. She was doing more. She was rolling over, and it was kind of almost like the calm before the storm started coming in. So if you can go into the next slide for February. So you can note the year, February 2020. We get into a lovely COVID nightmare. So at the end of February, that's when we had her MRI. But in mid-February, Alana stopped tolerating feeds, and she was hospitalised on that 23rd of February 2020. That's when things really started getting hard. I was told that the bloods ruled out any fatal causes of hypotonia, and I was thinking, whoa, I did not know we were looking at terminal issues here. The bloods were redone, more examinations. The MRI was complete. It showed she got small corpus callosum, benign hydrocephalus, and she had a few cysts. And she was hospitalised at the end of February for 2 weeks. She lost the ability to feed. She lost the ability to roll over. Every time she was fed, she was vomiting. They did manage to get her on a gentle feed at a gentle rate, and then she was discharged early March because the pandemic was coming in and it wasn't seen that she would be safe at home. If you can go into March slide, please. Next slide. So there you go, you can see her. So she's got her MG was in. There's a couple of hospital photos there. So we had tests booked in for the end of March to do with her vision. I was worried they'd be cancelled, but fortunately we did get in and a scan was a brain, the cortical vision. She had cortical vision impairment because basically the brain just couldn't pick up any. She wasn't even seeing any light, so it wasn't registering her brain. So we knew there was something wrong with the visual cortex there. By this point, we were put in touch with the community nursing team, but they couldn't come in the house. We needed support, physio, vision specialists, but again, none of that could be accessed thanks to COVID and lockdown. Alana had kind of, she was less sleepy, but when because she it wasn't the early days where she was like sleeping all the time, but she would scream for 72 hours at a time every 5 to 10 days. And it was, honestly, it was exhausting. I don't know how we got through it. If we can get on to the next slide, please. So we're in April there. So we did, because of this constant crying, they did actually manage to get, the rapid response team finally managed to get Alana admitted to the Children's Hospital. And Alana and I were descended upon by the neurometabolic team. I crumbled. I absolutely crumbled because of my background. And I was just like going, I knew if it was neurometabolic, I knew this is essentially a death sentence, how long she got. And especially as she'd lost some of the ability. So I could see what they were looking for. I think there were some markers which suggesting Krabbe's disease. So I said to them, I said, what is her prognosis? And they said, we can't say because we don't have like, we don't know what it is. And I said, but given the way she's presenting, and you think it's neurometabolic, how long would you give her? I know that, it's not good. And they said that it'd be unlikely she'd make it past 2 years. So I was locked in the hospital with my daughter with this news, with my husband and my 4-year-old at home, and I had to deal with that bombshell on my own. And it took days to get my husband to discuss a potential prognosis to get him in. But because of how poorly she was, they decided to do the whole exome sequencing on her at the time. It was relatively new at that point in 2020, and only critical cases could be run because the labs were all wrapped up in the COVID testing. So I knew it was serious. It was pretty devastating. So, and then we move into the May. So if you can do the next slide, please. There's me having my Britney Spears moment. I ended up shaving all my hair off because people were moaning about not being able to go to their hairdressers. And I was like, I've got a child who I've been told isn't going to live to be 2. Get some perspective. So anyway, we did get a call mid-May. We had a virtual meeting with a geneticist and a neurologist, and we got the daughter's diagnosis via video link. It was hard having it by a video link. But I don't think all the professionals there, as I think normally you'd have more paediatricians and things up there. So the fact we only had the geneticist and the neurologist, it was could have done with I think a few more people there. We did discover that her condition is actually neurogenetic, not neurometabolic, which was much reassuring because we were told that she could have a near normal life expectancy. It was such a relief to get that diagnosis. We were looking forward to getting some counselling after. However, that was just a phone call because of COVID. And I'll be honest, when I got to that counselling appointment, I'd already done a lot of the research about Mann syndrome. So it was almost like we got the horse after the cart. I don't know how it's done now exactly. I do think there is more scope for counselling now. I think it's been done before the genetic testing rather than after, which is good. That's a really good improvement because, as I say, I got I got it kind of, as I say, after and I knew more about it than the geneticist by that point. So since then, though, Alana is a lot more fragile than literature would suggest. We suspect she might have another pathogenic variant that's not yet recorded, or another which could be relatively rare. Just because she, as I say, she's been in hospital now 18 times this year. She's regressing. I don't know. So we just think there might be something else. In terms of her immunology as well, they're running whole genome sequencing panels for that because of just how poor she is, and yet she doesn't elicit a CRP response or anything when she's in. She's had sepsis with a CRP of like 30. She's had sepsis with a CRP of less than 1, so she's not producing a proper immune response to things. So something is probably going on. Something else is going on. That's the beauty of the fact that they keep rechecking. So if you can move on to the next slide, please. If you just want to get all the points up, I think there's because obviously I did it as clicks, so it's pretty easy. There you go, thank you. I think there's 4 points with something underneath. Yeah. So I know my story is going through the diagnosis process. It was a pandemic in the middle. I can tell you that there were some funny anecdotes of blasé things that have been said to us over the years. I don't think there's a right way, but I know there's a few wrong ways, things to say. Now, luckily, I'm quite, I'm the daughter of a policeman of forensic science, so I've got quite a dark sense of humour. So I brush a lot of things off anyway. But I can think, oh yeah, if that was somebody else, you might get yourself in trouble there. So the first one is that subject of consanguinity and child paternity. I know it's important. I know you need to find any genetic leaks if there's any recessive disorders. And I know testing will show the paternity of the child if there's any doubts about paternity. There's that ethical obligation. I get that. But you don't need to labour the point. So I ended up, every appointment I ended up going to, I was like going, right, this is Alana, these are her symptoms. My husband and I are not in a consanguineous relationship. Yes, I am sure he is the father. So I was giving that spiel before I could even be asked, because I really genuinely got fed up about being asked it. So I don't know if there's a way to have this information before the appointments. Might be easier, especially with the stigma for the consanguinity. Patients can sometimes feel that they are being blamed for something wrong with their child, and it's not the case. So it would be, I think, prudent to not keep harboring on, the parents feel guilty enough as it is trying to find out what's wrong with their child. But we'll always blame ourselves. Another sort of story on the second one is Alana's first ophthalmology appointment. The doctor wasn't really very professional. He's one of the only ones I'm really not that impressed with, because he started Googling in front of us, eye problems child. Now, my husband and I, we are both professionals. We understand you don't know everything and you can't remember everything at the same time. But don't Google eye problems child in front of the parents. I kind of would go without saying. And using Google of all search engines, you're just thinking, yeah, this is not filling us with much confidence here. Moving on, difficult one is that's of faith. Some people have faith, some don't have faith. I do, my husband doesn't, so it's like kind of one of those kind of, I don't think it needs to be brought up in this sort of setting, like a clinical setting, unless it's like kind of like, unless a patient brings it up first. So I mean, I've been told I've been chosen by God, or God only sends special children to special parents. Some might find this comforting. I don't actually believe that, even though I have got faith. I'm just thinking something happened genetically, unfortunately, is one of those things. My husband goes mad because he's like, how can that, like, why would that happen? It's not really that comforting. I think, unless the parents bring it up, you should leave it out of the clinic room. And then another one is kind of basically is patient history. Think about the language being used. As said, parents, we blame ourselves for everything. I remember going through, what did I eat? What did I eat in pregnancy? How much exercise did I do? Did I not do enough exercise? What lifestyle choices before I even got pregnant? Did I party too much in my uni days? All of it was going through my head. And then I agreed to have like a planned cesarean because it was what was recommended for me. But I was basically told, why did you choose to have a section? And I'm like, I didn't choose to have a section. It's almost like you blame yourself then. Go, oh, okay, so we had a section earlier than 37 weeks. Is that what's gone wrong? Is that now my fault? Why did you choose that? It's that language. It can be quite upsetting. And then another one around language is, you wouldn't have her any other way. And it's like, yeah, okay, I would have her. I'd have her healthy. Thank you. I don't understand why people would say that one. We can go on to the next slide, please. If you just want to get up all the points, because I've gone through each one. So one of the things is like minimising the conditions. Again, I had So one doctor did pick her up and says, "Oh, she's got nothing." And you're just like thinking, yeah, that's not very kind. We know, but like it's thinking, just again, be careful our language. I've been told like kind of you're lucky you know so much about genetics and medicine, and I'm like, how is that lucky? How am I lucky? If anything, it's worse because I know what's coming. I don't like it. It's not lucky. So that was like you got to be careful. It's next point. If you can put the next point up as well, and then it's like it says you can misjudge that emotional impact. So yeah, saying you're lucky to know so much, it ignores the burden that you're kind of getting as well. So the next point is delivering serious news in writing. No words of a lie. Because we were told, you know, from when we were told, like, okay, yes, she's got a near normal life expectancy, we then know that she obviously is regressing. We actually had a letter that come through last year stating children with Alana's degree of neurological complexities tend to die at an early age. No one has ever told me that in person. And I'm like, wow. Yeah, make sure that before you send out letters, you have had those discussions of prognosis with the parents. It's not great. And then another point is insensitive questions during subsequent pregnancies, of just being like blunt phrase of things. You know, it's like I do have a child, a third child, a little boy, who's younger than Alana. So we found out Alana, she was de novo. Me and my husband didn't carry it. It was just one of those things that happened at fertilisation. So, and we didn't want our older child to be the only healthy one, so to speak. So, at 7 months pregnant, I was told, "Have you had it tested?" And I was just like thinking, I laughed. I was going to say, "Yeah, I have," but it was done in a corridor, and there were other people around, and I'm just like thinking, okay, you could. There's better ways, better times to say that kind of thing. You can go on to the next slide, please. So making assumptions based on profession, not helpful. So again, I was basically, oh, that's what me and my husband do for a living. We told them, and I went, oh, good genetics. I just burst out laughing. Yeah, not really, because she's got genetic disorder. I did find that quite funny, but again, yeah, it's just something that not necessarily should say. The next point is ignoring parental expertise. Parents know their child best. I actually even had this week. Alana was in hospital at the beginning of the week, and I was basically saying, "Oh, this is Alana's baseline." I'm like, "No, it's not. You don't know her. You're a new doctor. This isn't her baseline. I wouldn't be in hospital if it was her baseline." I mean, she was back arching, screaming in agony. She was vomiting, having seizures. I'm thinking, this is not her baseline. So don't forget that even though a patient or family may have no medical background, or even if they do come from a medical background, they are experts on that condition. They live it. So, you know, listen to the parents, listen to the patients. And the final point on this one is like treating parents as professionals. They want to be caregivers. So it's the last point. So just having that kind of little bit of empathy, really. You know, yeah, I would say I am quite a tough parent. You know, I am quite resilient. I'm quite knowledgeable. And I think people forget that I am a mum. And although like it looks all calm and I'm like, yeah, inside I am like shaking. I am like worried. I am nervous. It is my baby at the end of the day. I mean, I've had, but like during lockdown, I was asked to do full diagnostics of whether she can track light and movement and things. I think I'm a mum. You know, I want to be able to look at my daughter as an adult and not as in a clinical way. So we aren't professionals, but we have become them. It's a quite difficult road to navigate. So things what made a difference. So it's not all doom and gloom. I say I'm very thankful for my wonderful, you know, some of the life consultants are amazing. So, so next slide, please. So how to get it right and what made a difference to us. I can only say what helped for me. I don't know everyone else, but like one wonderful clinician asked me about Lana simply flipping it. So, oh, what can she do? And I was like, oh, that was really nice, because you've always been told like kind of, oh, what can't she do? It was like kind of nice that it was flipped to sort of looking at her individual rather than her disabilities. That was really lovely. It's like highlighting her strengths and anything else. The next bullet point is, I say, be early clarity of what you're looking for. I do kind of wish someone had just kind of told me that they were looking for genetic orders early on. We know something's wrong, preparing them, preparing them for that. We understand, we've brought them there, we know something's wrong. So it kind of gives them validity that you are, oh yeah, okay, something is wrong, can be wrong. So it's like, listen to them, document them, don't brush them off. Make sure to document them, all the different concerns that they are coming up with. Next slide, please. So involve families as much as you can in decision making and discussions, but also remember, at some point you need to be the parent. You need to go into nurture mode if needed. And it's a fine line to don't dismiss them, but at the same time, you are still, you are the professional, so it's like quite important that you do push if a certain treatments are needed and things. So being as supportive as you can, offer some practical support. So giving contact numbers, follow-ups to secretaries, contact numbers of the secretaries if they have any queries. Try and go for the operators. It takes forever. So even if you can just, if you've got a direct line to the secretary, so that we can, you're being pointed in the right direction. And then show empathy. It's essential in any clinician role. I think that's why a lot of you do go into the medical field. But remember, parents are going through a grief process. No parent envisions having a disabled child. They're grieving that healthy child they don't have. It doesn't mean that they don't love their child, but it comes in waves, this grief. It's like sometimes parents accepting, and on their appointments they might not be, especially if there's been a lot of hospitalizations. It's that constant grief. As I say, it's a grief process. You're grieving for someone who's alive. It's a very strange place to be. So onto the last sort of slide. This is where Alana is now. So this was her over the summer. You can see she's actually a happy little soul. There's one of them in hospital there, but there's a daddy and her brothers. I say when she's healthy, she's a wonderful little girl. So thank you for listening to my story. This is just my experience. Everyone is going to be different. I can't speak for everybody. But yeah, that's it. So has anyone got any questions?
If you don't mind, Cara, we would normally just shift to the end for questions, if that's okay with you.
That's fine.
Right? Wonderful.
Okay.
Thank you.
Lovely.
Thank you so much, Cara, for sharing that. I know it's so difficult to talk about this stuff sometimes and these kind of experiences, but it really is just, incredible to hear you share your story and some amazing advice and tips there, I think, from somebody who's been through this process with your family. So thank you. I am going to hand over, and like I said, if you have any questions, we are going to do a Q&A at the end where you can ask any of our speakers questions, and you can pop them in the chat if you don't want to forget right now. I am now going to hand over to Helen Bulbeck, who is speaking on her experiences as a caregiver on humanising the process. So Helen, do I have you on the call?
You do. I'm here. If you could go to my first slide. I don't know how to silence the chat that's coming up, so just bear with me. Do you want to go to my slides? Thank you. So my story is a little bit different in that I'm a generation apart, and that was an amazing story that you shared, Cara. I'm a patient advocate on the Genomics England patient participant panel. My journey with Genomic started when my daughter was diagnosed with a brain tumor when she was 19. So I'm going to tell her story. A lot of it is in her own words. She's now a psychotherapist, and she gave this talk at the European Association of Neuro-Oncology. And it's about how having a genetic diagnosis is really the gift that keeps on giving, and it rolls down through the generations. It's not a defined moment in time. You get told the diagnosis, but it lives on through the generations. And that was the one thing I hadn't considered when we were told my daughter had NF1. Meg was diagnosed 20 years ago. She was 40 this year with a grade 2 ganglioglioma, which is very rare because gangliogliomas are normally grade 1. You can live with them for a very long time. She was diagnosed in Southampton following a seizure when she was 18. She'd had a really good upbringing. She was born at 34 weeks. I had a placenta previa with her, which was abrupted, so I hemorrhaged. And she was an emergency C-section. A lot of that pregnancy I was on bed rest because we knew I had a low-lying placenta. When she was born, she was covered in loads of cafe au lait spots, and I just thought they were exquisite and beautiful, and they were part of Meg. It never occurred to me that they could be an indicator that she actually had NF1. Her symptoms started when she was about 14. She'd wake up being sick. She wouldn't get up and be sick. She would wake up being sick. She lost a load of weight. And then things gradually started to go downhill, but obviously being really naive about these things, she said she had a flashy eye. I was a migraine sufferer, so I just thought she had headaches. And that's what culminated in her having a full-blown seizure when she was 18 and ended up in A&E, which prompted then the MRI scan and then the brain tumor diagnosis. If we could go to the next slide, please. She was off to university at this point, and we were just managing the seizures, and she wasn't interested in her brain tumor diagnosis. She just wanted to get on with her life. And I think out of the experiences, I was then diagnosed with head and neck cancer a year later. And out of that, Brains Trust grew. So I now run a national brain cancer charity. And the one thing I've learned is that before we're patients, we're people. And it's very easy, even when you are the patient, to lose that perspective of yourself as a person. And I think it's really important in the current climate. You know, we've got the thrust of integrated care systems, which leads towards a system-wide outcome measurement. But there's a case that this also loses sight of what is key to this, and that's the person living with the disease and what matters to us. Because there is no story to be told without the patient, and we are the only form of data. And the value that we can bring to the agenda exceeds that of the purely clinical. And I think the genomic sequencing add colours to the other data sets that which are awesome. So the diagnosis, the biological, the environmental and the behavioural influences that we're living with. It means you can match the right therapy to the right person at the right time. And also there's the health economic argument. And this leads to a better management of expectations. So patients like us will be able to see how others are experiencing, what they're experiencing compares with our experience, and we can become co-pilots in our care, improving our experience of care. And having us as co-pilots reframes problems, it can generate insights, and it shifts dynamics and changes practice. So to cut a long story short, the first neurosurgeon we saw walked into his room. It was our very first consultation. And he just said, I'm the best neurosurgeon in the south of England. And before I could stop myself, I said, well, I don't think that's a very big place. And it kind of raised some questions in my mind and it took me a year to get to the point where actually I thought they don't know any more than we do at this point. And it was a completely serendipitous conversation that I had that involved a rabbi in Israel, and all the time I was thinking, this isn't real. I'm not going to go down this route of listening to this rabbi. But anyway, he's Rabbi Fira. He has in his head a database of the best clinicians in the world. And he recommended 2 neurosurgeons in the States. So I just had a scan sent over. And that was interesting too, because when I took the scans were film at this point, not on a disc. When I took them out, there was the neuropathologist report that said that this was a grade 2 glioma and it would transform to a highly aggressive brain tumor within 5 to 7 years. And by now we were kind of 2 years beyond her diagnosis. She wanted to get on with her history of art degree. So we sent her scans to the States, had a chat with the neurosurgeon. If we could go to the next slide, please. And we ended up going to America, which was the one thing I vowed I would never do, because I've always had faith in the NHS for her to have brain surgery. So the day after her neurosurgery, the professor of neurosurgery at Harvard and the anaesthetist came to us, and she obviously was under lots of observation. She was in ICU. And what she said was that one said that his colleague had noticed he had lots of freckles on her tummy, and Meg said, I felt utterly exposed, looked at and talked about. I nodded, she said. Do you have them all over, in your groin and your arms? Yes, I said, I'd always had them. The doctor was excited and beckoned his colleague over, as it seems to me these freckles, and then what I learned, were actually cafe au lait spots and neurofibromas, a markers of neurofibromatosis 1. And because they had her tumour in their lab and it was a rare tumour, they were pleased that they'd been able to use it for their research. And Meg was happy that it could be useful to them. But she really didn't feel like a person at that moment in time. She said she felt like a specimen. The freckles that she'd never thought much about became something horrible and sinister. And then further testing on our return back to the UK, she had Lish nodules in her eyes, another marker confirmed in NF1 diagnosis. Next slide, please. So time moves on. Meg married and wanted a family, and she and her husband went through the genetic counselling. These are her words in the slide, and these are her words. We all have an intense and overpowering parental urge to protect our children. Meg wondered if it was selfish to have children, if there was a chance that she could pass NF1 on. And given that this faulty gene, this mistake of nature, this error had occurred within me, what would I be the starting for generations to come? But on the other hand, is the non-existence of children a better option? I felt not. My husband was adopted, and that was another factor in deciding that we really did want biological children of her own. So Meg had 2 very easy pregnancies and 2 gorgeous girls. And at the end of the 6-week check with the youngest, the GP noticed something unusual in Audrey's eye. Meg found it irrationally hard to connect to Audrey because of this. And she was convinced that she'd passed NF1 on to her. And she said she felt unable to love her children fiercely enough as she should have done, because she was sure that something terrible would happen to her, that she'd have some awful malignant tumours and it would all be her fault, and that she'd be living with this diagnosis for the rest of her life. So if you go to the next slide, please. So the emotional cost of living with this diagnosis. So Meg's been living with it now for 20 years. And it was a real eye-opener to me when she stood up at this conference and shared this information, because I had no idea of the ongoing impact of this diagnosis as her mother. Her last scan was in February this year, and I always go with her. We followed the consultant in, and he spent some time looking over the scans. While he was looking, Meg was imagining that he was seeing tumours everywhere in her brain, but after what felt like a long time but was in reality only a minute or 2, he said it was excellent news, her scan was stable, there was no change, and the consultant asked kindly if she had any questions. Meg says, my only question was to myself, why do I want to cry? I shook my head and said, no, thank you so much for seeing me, thank you for your time. I avoided looking at him so he wouldn't see my eyes reddening and prick, worried he would think I was silly or hadn't understood the brilliant news that my scan was stable. I hurried out of the consulting room knowing I was about to burst into tears. It was confusing. There was no whoosh or feeling able to breathe again. My breath is still held. In that moment, the years fell away, and it was as if no time had passed at all. I was right back to the moment of that initial diagnosis, and that's the reality of living with a genetic condition. I realised that having NF1 has affected me in more ways than I knew. I watched myself parenting, terrified that in normal moments I imagine we all experience, when I'm bored, frustrated, wishing to be elsewhere, that I will pass these bad feelings to the 2 gorgeous girls that I have and ruin their precious hearts and minds. A fear of passing things on emotionally that stems from the reality of my fears of passing on NF1. I've wasted years feeling like I'm on borrowed time, robbing myself of precious moments with my daughters, because all I could think about was not being here anymore. I've had times of checking locks, doors, windows repeatedly, almost obsessively, until I understood that I wasn't afraid of not being safe in my own home. I was lacking safety in my own body, because not just of my fear of recurrence of my brain tumour, which is always on the cards, but my fear of other primary tumours developing due to NF1. This is all irrational, I know, but feelings are irrational. Next slide, please. She then talks about the emotional benefits of having a positive genetic test, and learning the results of a genetic test that your brain tumour is or is not hereditary can have a significant impact on one's emotional well-being. It can be a relief and end uncertainty about one's gene status. It can put an end to the not knowing, and often it is the not knowing that is the hardest to live with. It may clarify risk for other family members, and it can impact medical management decisions such as taking up screening, which can give a sense of control back. But the emotional risks of a positive genetic test, if we could go to the next slide, please, are far greater. Research shows that people do not regret undergoing genetic testing for hereditary syndromes, but reactions can be more complicated than they or you anticipate. It can increase anxiety, sadness, and hopelessness. Some anxiety is expected and can be useful in motivating patients to attend appointments and scans. Too much, though, can be freezing. It can make a patient withdraw and avoid appointments. It can also be cyclical, increasing around the time of scans or screening, and strain family relationships. There may be guilt from the burden of the family of being unwell with hereditary syndrome, or in Meg's case, a low-grade glioma, or guilt that comes from concern of passing the hereditary syndrome on. There's a shift in identity, which is pertinent to brain tumour patients anyway, as it can bring changes in physical and cognitive capacity, dependency on caregivers, and loss of independence. And a diagnosis of hereditary syndrome can also increase the patient's sense of identity, being based around medical diagnosis and added vulnerability. There's a deep-seated feeling too that there's something wrong with you, that you're inferior or unworthy, or feeling uncertainty in the future as the hereditary component makes things less certain. And then the isolation that comes with having to live a life that is distinctly unique from family, friends, and colleagues. Even clinicians can be unfamiliar with hereditary syndromes and brain tumours, and can feel like no one understands your worries and concerns, which can make you feel unsupported and unheard. And then the shock, as the trust you once had in things might shatter. It's a disorientating feeling. So if we could go to the next slide. And this is, more my perspective on working with people living with brain tumours, some of which do come about from a genetic disorder. This is the zeitgeist. So we understand the issues around prompt diagnosis, not early in brain. That's another presentation. But the nature of rare diseases, that means unless a healthcare professional specialises in the area of medicine, it can be a struggle to get a prompt diagnosis. Patients can have multiple visits, see a variety of healthcare professionals, making the pathway difficult to navigate. Primary care may be the first port of call, and the GP may have limited knowledge. And I think that was very much reflected in Cara's amazing talk. It's important that healthcare professionals have a working knowledge of genomics and its application to clinical practice. And you have an ethical obligation too to explain the risks, the benefits, and the alternative of an intervention through shared decision making. And patients need time to process this. At the point of diagnosis, you have to absorb a lot of complex ideas very quickly. And having the genetic analysis doesn't mean that there will be a therapeutic intervention. This needs managing too. Currently, very few genetic diagnoses have approved treatments, and support must be available, signposting to trusted information, and tumour-specific support groups, particularly relevant with less common cancer. I think patients would expect that a multidisciplinary team would be involved, and that the team is suitably prepared so that the patient doesn't get the results in a chaotic manner. This is harder to manage as we have access to our patient records online. It's not enough to provide a complicated report, it needs mediating. And we have to live with our conditions 24/7. This enables us to be world-leading authorities on our diagnosis. Our voice is essential and central to planning, research, and care pathways. We can bring patients closer to research and research closer to patients. This is particularly relevant when you are part of a less common or rare disease community. The provision of real and virtual communities helps to shape and influence research and policy. We're an awesome force, so please use us. And these, next slide, please. These are the considerations for people who are living with a brain tumour. We'd like to grow the conversation. There are focuses that the brain tumour community want to understand and deal with. So what does the average patient need to know to be able to make an informed decision about their diagnosis? What does this particular patient need to know and understand to be able to make an informed decision? And how do we close this gap? And that's a lot of the work I do currently in Brains Trust. But next slide, please. To be able to close this gap, these are the things we need. But of course, there are headwinds to this. There's a lack of understanding about how research funding needs to be spent. Equity of access to whole genome sequencing is an issue. Too. Timing. It was 411 days for somebody who was diagnosed with a glioblastoma. Well, given that a glioblastoma, it's the most progressive of malignant brain tumours, you're looking at 9 to 14 months. The genetic sequencing was coming in after the person had died, but it has speeded up. We need drug delivery trials. We need trials, full stop. There's a lack of infrastructure with the NHS, with whole genome sequencing, human resources and time. Tends to be a focus on cures and treatments rather than quality of life. Access to local resources too, and then mindsets, the human condition, the shortage of resource and funding. Next slide, please. These are Meg's words in the slide. As from the patient's perspective, it's about developing agency in people living with disease so that we can become co-pilots. But to do this, we need the tools, the networks, and the license to be people first. And this is going to take a considerable investment in technology, new tools to aid decisions, and a change in culture for everyone. And genomics needs to be in the DNA of this transformation. Next slide, please. I'm very nearly finished. This is the last one. It comes back to the power of the narrative, so telling a story, and Kara's story is just hugely powerful, eliciting from the person living with the disease what matters to them, what their values are, their context, their appetite for risk is a transformative conversation. And it's those types of conversation that place us as co-pilots in our care, where we can be regarded as an asset with our own sense of resourcefulness and range of capabilities. We'd be more likely to engage with research, we'd be more confident and better able to navigate our clinical pathway, and we need to be able to recognise this as a person-centred process so that quality of life can become just as much a rich theme in research as a search for a cure. And we'd also have a good story to share. There's some references in the next slide, and then my contact details are on the last slide. So thank you. That's my presentation done.
Thank you so much, Helen. That was really, really informative, and it's really good to hear. Well, not good to hear, but it's really great to hear the story and how you've worked through, and obviously all your work with Brains Trust. So as I've said, we will send these slides out, so you'll be able to access all of these links, and we'll share some information on Brains Trust as well in the follow-up. So next we're going to be moving on to Pauline O'Connor. So Pauline, do I have you on the board?
Yes, I'm here. Hopefully you can hear me.
Yes, I can hear you. Wonderful. So I'm going to hand over to Pauline, who's going to talk a little bit more about the sort of pharmacogenomic treatment option side, which we cover extensively in other webinars, but it's great to hear that from the patient perspective too. So over to you, Pauline.
Thank you. Yes, those were two very powerful presentations. Mine is going to look less at the sort of diagnostic journey and more at how genomic testing might help with future treatment. So do you have my slides there, please? So yeah, my name is Pauline O'Connor, and I was actually diagnosed at birth with a rare disease. So I have lived with PKU or phenylketonuria for over 40 years now. And that is diagnosed at birth. It's through a blood spot, or it used to be via a nappy test that happened with children. So some of you might recognize that. So it's diagnosed through blood spots rather than genomic testing. But I went through genomic testing as an adult, looking at a possible new treatment for PKU. And just to let you know that I have had sort of I've got 40 plus years of living with a rare disease, and my parents were both in the medical professionals, and some of my family have had unrelated chronic actually brain injuries. So I've had extensive experience both as being a patient, being a family or caregiver for someone else. So that's where I'm coming from. If we could look at the next slide, please. A little bit about my rare disease. PKU is sort of the oxymoron. It's one of the most common of the rare diseases, and it is a metabolic disease. So it's through, I believe, a missense in the genome, going back to that first presentation. And people with PKU, we are very lucky in the sense that we have a diagnostic test, which is part of the newborn screening program, and there is an existing treatment for PKU. So that's quite unusual in terms of the rare disease. We don't tend to have that big diagnostic journey. However, the treatment is over 70 years old. Going back to Dr. Tetheridge's slides at the start, when she showed those cars that were from the 1953, that's actually the same year that treatment for PKU was developed. So I am currently on a treatment, but I'm on a treatment which is 70 years old. And the treatment is severely restricting protein intake, because my body basically cannot produce the enzyme that breaks down one of the amino acids in protein. So the treatment that was developed and the treatment that majority of people that are on is a heavily restricted diet. And we are only allowed 15% of foods. So I've never eaten meat. I can't eat meat or fish or shellfish. And it's not as simple as a vegan diet because I can't have chickpeas. I can't have tofu. It's incredibly restricted. And that means that it's quite an exhausting treatment because a lot of our social activities are based around food. And to go back to the biblical story of, you sit down with someone and you break bread with them as a way of connecting. I actually just can't eat bread. I mean, that's another fruit. I can't eat bread. So that becomes quite an isolating treatment. So we are lucky that there's a treatment, but it is not an easy treatment to follow. It takes willpower every day. So if we head on to the next slide, that brings us to the genomic testing, because in the last couple of years, there have been new treatments developed, and one of them was approved for use on the NHS. Now, this drug, it boosts the enzyme activity in some people with PKU. So it works in about 30% of people. And essentially what it does is it takes the existing enzyme that the person produces and it boosts them. My incredibly unscientific understanding is that in some cases it helps to refold the enzyme in a way that helps it to work. That could just be me speaking gibberish. But essentially, whether you respond to this possible new treatment is dictated by two of the inherited genomes that you get, which cause PKU. And so far, we know of about 1,000 different mutations involved in PKU. So one person's PKU is not the same as another person's. And that is because there are all these different variations in the type of PKU someone might have. So that's why I had genomic testing as an adult. And why we do genomic testing is because we need to find out whether this possible new treatment will work. So if we skip on to the next slide, just to take you through how my genomic testing went, it was incredibly unlucky timing. It was during the pandemic, or as the NHS was, or actually still is, I think, recovering. So the drug was approved at the end of 2021. And my clinic was very good. They were on top of it. I was contacted almost immediately. I went in and got the blood test and I was terribly excited. It was my first time in my entire life that I was possibly going to get a new treatment. And as you can see there, the difference between expectation of results and how long it actually took was vast. The system was completely overwhelmed. And it's understandable given the circumstances. There was a lot of recovery from, the emergency mode that the NHS was in. I believe there was also some problems with getting some of our experts over from the EU because, of course, Brexit fell in there. But it took an incredibly long amount of time. Well, it certainly felt like it. I'd waited 40 years and 6 weeks to 7 months. It doesn't feel like a lot, but it is when you have to live it every day. The actual communication of the results with my clinic was great. They gave me a call. They asked one of the key things they said was, are you able to talk with us about this? Because that just let me know that it was time to leave my desk and get into a quiet place in the office. I think that's a very good thing to lead with, because you never know whether someone's just answering the phone during a bathroom break or something. So that would be a top tip, is just when you call someone, just tell them that, give them an inkling that you will need some of their time and attention for a while. And then it followed up with a letter, so I had all the information, and actually, despite the long wait, the communication of the actual results was really good. So if we head to the next slide, and that will tell us that, yes, unfortunately, my results were not what I'd hoped for. One of the phenotypes was known to be not responsive. And the second phenotype, and this was quite key to my experience, the second phenotype was not known. So it wasn't one of those 1,000 known variants. And the letter actually stated, can't predict the responsiveness. So the end result was, I was unlikely to respond. And it was pretty heartbreaking, actually. I'd waited a very long time, and it came back as a no. You have to keep doing this same diet. There's not going to be any change or excitement for you. So yeah, I did cry, and then you sort of get on with life. And so that sounds like a very simple process, but if we pop onto the next slide, you'll then see that things got a bit difficult, because there was sort of confusion across the country. Sorry, if we pop onto the next slide, please. Because it turned out that across England, because it was only available in England at the time, people were treated quite differently given their different clinic results. So my clinic had said I was not going to go on and complete the drug trial, whereas I found out that other patients who'd had the exact same responses as me. So one phenotype was known it wouldn't respond, and another phenotype was completely unknown, but they guessed it wouldn't respond. Their clinic had said they could go ahead and do the trial, and indeed some of them did respond. So that really caused a lot of, well, as it says, it felt really unfair. And what I'd initially had is extreme understandable disappointment, that I would not be able to, I mean, this new treatment allowed people to eat a vast amount more food than they had been allowed to before. Some people were able to eat an almost normal vegetarian diet. We're talking very life-changing treatment. And I felt like because of my postcode, I'd been told it's not going to happen. So what was understandable disappointment turned very quickly into anxiety of, should I have been advocating more? Should I have asked more questions? Was there some kind of password of medical jargon that I could have used that would get me into this secret set of people who were allowed to try this drug? And all of that could have actually been quite easily avoided, had there been a nationwide plan across the clinics of how are we going to treat the different results. And that did not happen. So it should have been quite easily avoided. And if we pop onto the next page, I was thinking about this because there was actually a lot of ill feeling as a result of the process because of this. And my rare disease, it's rare, but it's common enough that we have a community. And I really felt a lot for the clinics, because one clinic doesn't necessarily know what another clinic is doing, but we meet at conferences with other patients and we know what other clinics are doing. And I think had there been any kind of thought beforehand about, clinics are not bunkers, patients talk, then life would have been a lot easier for patients, and certainly for metabolic clinics. They would not have been faced with a lot of the real anger and disappointment that I think came about because there was no consistency. So that was my first sort of feedback on what to think about when you're doing or putting together a sort of genomics testing program is, do take the time to sit down and think about what are the different types of results that we could get. And for each and every result, whether it's going to be a yes and a yes, no, or yes, yes, or, you know, if it's going to be an unknown result, have a consistent answer that you give your patients. Even if the answer is, we don't know, we're taking your results, we are going to look into this and get answers for you. Just having that consistently said across all your patients will just help you and your patients to deal with the process much more easily. So taking that time beforehand, and particularly for ambiguous results like I received, and as we all know, genetics are fun, they pop up all sorts of different answers. But just have a simple script that is, we've got your results, we've got your details, we will get back to you as soon as we know what's going to happen here. And the second one that I still haven't been able to find out exactly what's happened to my results, because apparently there was an idea that because I had an unknown genome that they now know doesn't respond to this treatment, that was going to be recorded somewhere, so that if someone else has the same variation, they could then know that it's non-responsive. But according to the letters, that's never been set up. Or and when I asked my clinic about it, they still aren't sure what's happening there, and that's 3 years. So again, it's thinking beforehand about what's the process for the data as well. So you've got the process for the patient and what your communication with them is, and then your process for the data, what's going to happen, even if the data sits to one side until you've dealt with all the patients and then you've come back to it. Because really what we're talking about here today and what genomics, it's going to transform lives. And it's going to transform the access that patients and doctors have to new treatment. And the thing is, that can cut both ways. Patients' experience and doctors' experience can be both good and ill when it comes to genomics and pharmacogenomics. So it's really important to have some kind of consistency. So we pop on to the next slide, because I'm actually, despite this experience, which did, you know, it really knocked me for six. It was about 2 months before I was actually willing and able to talk to my parents about the fact that I had not responded to this drug, because that was how much we'd hoped for it. I am still very excited about the possibility of genomics and pharmacogenomics in treatment, well, in all treatments really. But we have to make sure that we do, you know, this, I call them the small things, because it's not as exciting as discovering a new rare disease or discovering a different genome or a new variation and how it how that responds to things. That's not exciting, and it's not going to get you a new paper. But we really need to take patients with us on this journey of genomics and pharmacogenomics. And that means we do have to do the small things well, the things like communicating. And it is okay to say that you don't know what something means, but you have to, you know, be able to say that and tell someone, we don't know what it means, but we are looking into it, and this is our plan for keeping you informed. And it can just be as simple as that. So I think there's a fair bit more sort of planning and communication that can go into it. And as with all things, you know, you adjust as you learn and you iterate along the way. So that would be sort of my experience of genomic testing so far, because these are really exciting times. And I think it's important to remember that what we're doing here with the start of these genomics programs, it is going to shape public attitudes in future. And I think that's where transparent communication is really important. So that was my whirlwind trip through my experience of genomic testing as an adult. So thank you very much.
Thank you so much for that, Pauline. It's really great to hear that you still have this hopeful, possibilities abound attitude. So thank you for sharing. Very powerful. I am now going to hand over to Anna Busby. Anna, do I have you on the call? Oh, sorry.
Yes, I'm here.
Sorry, I forgot to remove one slide. So Anna is going to talk about her family's experience of the sort of diagnostic odyssey, as we call it in genomics. This is sort of what we use, the term that we use to refer to the extraordinarily long pathway that a lot of people have to go through to get some answers. So Anna, over to you.
Yeah, thanks. I don't have any slides, so I'm just going to talk to our experience. So we have two children with two very rare, different genetic conditions. So the first occurred when we had our third child, Eli, in 2022. Eli was born at term. He was developing normally for the first 2 months. He did struggle with breastfeeding, which I think is quite common with children with genetic conditions, but we saw a really good breastfeeding counsellor and we got him on track. Then he hit 2 months and it was the bank holiday, Easter bank holiday weekend. We were out as a family having just a day out at, I think, in Bristol. And he started doing these funny kind of head drops. And I was saying to Joel, you know, he's being really odd. He was feeding and his head was kind of bashing into me, and I was thinking, God, this isn't normal. And they kind of occurred on and off throughout the day. And then later that evening, I was in bed and he started doing it again. And I thought, God, this is just not right. Something is not right. And of course, I Googled it because that's the first thing you do. And interestingly, one of the first things that came up was a Facebook page for infantile spasms, which I had never, ever heard of in my life. So I immediately joined the group and was accepted straight away. And there were loads of videos on there of children having seizures and having infantile spasms. And it looked exactly like what Eli was doing. So I think I must have watched videos for about 2 hours in the middle of the night. And then that was it. I just packed Eli up and off to the hospital we went. Because it was the bank holiday weekend, we couldn't get an EEG. But we did see a neurologist who has been our neurologist for the last 3.5 years, and we love him dearly. And he said straight away, yeah, I think this is exactly what it is. And we're going to start him on treatment now, you know, without having it confirmed. And we'll get that confirmed at a later date. So we did start treatment and it was successful and it stopped his seizures, which was brilliant. And as I said, he was only 2 months old. So in terms of development, we hadn't really seen any regression or any massive developments because he was only 2 months, but that was our main worry. So we went through the treatment process. It was a huge relief. We had an MRI done to check the structure of his brain. We had that done on the Monday morning. Monday late afternoon, our neurologist called us and said, yeah, no, it's great, it's all clear, nothing wrong with his brain. So, you know, we thought, oh God, huge sigh of relief. We had done all the blood tests for the genetic testing, but we were told it would take months for those results to come back. So we put it to the back of our minds and just kind of got on with our week, really happy that Eli's brain was normal, his seizures had stopped, and everything was absolutely fantastic. That same week on the Friday, our neurologist called me and said, I've got some news. It's not good. Can you talk? And I said, oh God. And I was on my own at home with Eli, my older 2 boys at school, my husband at work. And he delivered the news over the phone that Eli had his genetic testing had come back and it was an extremely rare genetic condition that was only discovered in 2017. So there wasn't much research on it, but he was going to send me the one research paper there was. He explained to me that it was not going to be what we had hoped for Eli. And obviously I was in tears. I managed to phone my husband. He came home from work. We read through this scientific paper, which made absolutely no sense to us whatsoever. The only thing that stood out was in severe intellectual disability. And that was all that was going through my mind constantly. Severe intellectual disability, uncontrolled seizures. And it was just heartbreaking. So then I needed help. I didn't understand what was going on. I don't have a scientific background. So I spent the next 2 weeks attempting to get answers, get an appointment, which after hounding people for a week solid, we managed to do. So we went to go and see our geneticist, who's a lovely lady. But my experience of that appointment, I had to go on my own because my son had to have the other 2 children. My experience was really awful, and it was heartbreaking, because although we'd read the words severe intellectual disability, it hadn't really sunk in yet. And I was there with my sister, and one of the first things that the geneticist said was, oh, it's good news because it's a genetic mutation. So you haven't done anything, your husband hasn't done anything, there's nothing you could have done to prevent this. It just occurred, you know, on conception. And I thought, oh, great. And he said, and the other good news is that it won't be passed on. So it wouldn't go down to Eli's children. And the words that really hit me was, she said, not that he's ever going to have children. And that was the moment I just absolutely broke, because I hadn't thought, sorry, it still gets me now. I hadn't thought that far into the future. We were still just like, what does this mean? What does this mean? And the idea that we're talking about a 3-month-old baby now, or a 4-month-old baby, was being told, right, that's it, you're not going to have children, it's not an option for you, was kind of the most devastating thing for me and to have it delivered in that way was just absolutely heartbreaking. And I think I left that appointment and just for the next few weeks couldn't really do anything. I couldn't function, and it was just awful. But I wouldn't change getting his diagnosis that early, because it opened up so many windows for Eli. So within a month, we had seen a paediatrician. He was having physiotherapy. We had an occupational therapist. We had a play specialist coming out to the house 1 day a week. And he's had access to all of these services for the last 3.5 years. And they've been amazing. And these professionals know him so well. And they help us all the time. So it was absolutely devastating. And the way the news was delivered to us by our neurologist, who, as I say, we love dearly, over the phone, and then not having to fight for access to speak to someone who could actually explain this to us, and then having news delivered to us in that way, I would hate for anyone to go through that process again. So I think the delivery of genetic diagnosis really needs to be carefully considered, and if it needs to be delayed until someone who can communicate that properly, face to face, can do that, I think then it should be. Then move forward another couple of months. Our eldest son Enzo, who was 7 at the time, he began having seizures. We thought, oh God, no, not again. We knew that it was very unlikely to be the same as Eli's because it was a mutation, and Enzo up until that point had developed completely normally. He was talking in full sentences at 2. He was walking at 10 months. He was advanced for his age, but he had struggled a lot in school with reading and maths. And we also suspected he had ADHD because he was very, very hyperactive. So we got in touch with our neurologist, who said, yeah, no, I will take him on. I will see him immediately. And we went through the genetic testing again. Again, it came back really quickly, within a couple of months, and we had the all clear. Nothing wrong genetically. His MRI was clear. But we just knew that there was something, you know, we knew that Eli's genetic variant hadn't been discovered until 2017. We thought, well, this is something that they don't know about. He's got a genetic condition, but they just don't know what it is. So we had time to kind of prepare ourselves. And then a couple of years later, after we'd gone through more testing to see if Enzo was a candidate for surgery, our neurologist said there is one more test we need to do. It doesn't come out on the usual genetic sequencing because it's a blood test. So we need to take some of his blood, and it's called ring 20 chromosome, which is, again, it's rare, not as rare as Eli's, but still very rare. So I immediately got on Google again, found the Facebook page, looked it all up. I thought, he's got it. This is what it is. It was just down to every last detail. It was Enzo. So when we eventually got his genetic result back for this and it came back positive, it was a whole different experience because we were so much more educated about what it was. We'd had time to come to terms with the fact that actually we knew this was going to be genetic in some way. So it was two very, very different experiences. We still haven't spoken to anyone about Enzo's genetic diagnosis, which we got in January this year. We do have an appointment for April next year. But, for us, that's fine because we're dealing with it. And it was a huge relief to get Enzo's diagnosis because he has struggled with seizures for almost 3 years now. And we've not been able to get control. And it was almost a relief that we could finally say to ourselves, his behavior has regressed. He does have ADHD. So having all of these things validated where people have said, oh, he's just a hyperactive child, there's nothing wrong with him. It's been a relief. So for us, genetic testing has changed our lives completely. But we don't think we would do anything differently. And just going back to Eli as well, one thing I forgot to say was that the genetic testing, we think, actually saved his life because 2 weeks after we got his genetic diagnosis and I'd found a small community on Facebook, sadly, one of the children passed away aged 3. And the parent posted a message on the Facebook group saying that unfortunately he passed away due to aspiration pneumonia. Now, Eli had been breastfed up until that point, but because he was on steroid treatment, we'd had to switch to combined feeding with a bottle. And actually, the day I read that message, Eli had been wheezing a bit. And something in my gut told me that he'd aspirated on his milk, on his bottle feed. And so we got him straight to hospital, and that was the case. So I think if we hadn't had the genetic diagnosis, hadn't found that community, that post hadn't been put on Facebook, we may have just put it down to a cold and we may have lost Eli in a similar way. So it really has, it's put a huge strain on our family knowing these genetics, but it's also been absolutely wonderful because it's helped us in so many ways. But yeah, my main thing is think about delivery of news and support available. And that's it. That's it from me.
Thank you so much, Anna. It's really, really powerful to hear everybody's stories in this way. And I think we do some of this in the NHS, but it's great to have something that is just dedicated to doing this. So thank you so much for sharing your story. We are now going to move through to the Q&A. Now I'm sharing, so I can't actually see the chat function. Does anyone have any questions? I'm going to hand over to Hannah and Emma to kind of chair the Q&A session. But if anyone has any questions, please raise your hand or pop them in the chat, and Hannah, Emma, Tony will be able to take a look and see if there are any questions.
Thanks, Isabel. And thanks so much to everyone for sharing your experiences. It's been really insightful. And I know that everyone will be having a think now about some questions. But I wondered if we could maybe kick off. Anna, you talked about giving how you received the diagnosis, and all of you have talked about that at different points in your talk. And I wondered, for all the healthcare professionals maybe on the call, if there's any advice you'd give for sort of making sure that when they give you that diagnosis, how they can do it in the best way for the patients. don't know if anybody had any of you had any thoughts about that. And Anna, you mentioned maybe thinking about the sort of timing of it and whether you maybe plan that or rather than doing it over the phone.
Yeah, I think it would be really good to, and I know the NHS is so stretched, but to at least have an appointment with a geneticist so that you have someone to talk to lined up if they can't be the ones delivering the news. I think would be really helpful. Not having to wait and chase and do your own research online.
Thank you.
I think if possible as well, it's just the time as well. So I get that, you know, the NHS is stretched and it's hard when you've only got like a 10-minute, 15-minute appointment slot. If there's ways to have it like longer, so you can have that sort of build up that relationship with the parents. I mean, it's delivering news is always going to be difficult, but just trying to be kind of like sympathetic to the parents, listen to them, don't rush them, but also just be able to sort of explain things in more layman's terms if possible.
Yeah, thank you. That's really helpful. So just looking through the chat right now, we've got a comment here from Louise saying that something needs to be done to be reinforced during the education and training of healthcare professionals. So thinking about that sort of personal. Thank you, Louise. I don't know if anybody else had any questions. Yes, thinking about communication skills. Thank you. I don't know if anybody had any questions they wanted to raise their hand. Emma?
You did, I know at the end of your talk you mentioned, as well as delivery of the news, support available is something we should be carefully considering. And you mentioned the Facebook group, but were there other resources that you found helpful and you would maybe suggest to others that were really helpful to you, or anyone that was on the talk?
I think for me, because the particular genetic conditions are so rare, there's just not that much out there. There aren't any kind of official support groups, but the use of Facebook, I think I would say, you know, you get to speak to other parents who are going through it and who have what they've tried and their advice, and I think that's invaluable.
So that community.
The Facebook group for Man's syndrome as well. So like I'll get more support from them just to find out like what's going on. Unfortunately, Alana is more, I say, the most severely affected in the world, which isn't helpful sometimes, or seems to be for anyone who's in that community. But like I say, that they do become really useful. I know like Unique, they do have like kind of information on their website, but it could do with updating. Like it's almost like you get the genetic disorder, they find the genetic diagnosis, they're like, oh, this is what it is, this is the phenotype, but then they don't really update with how prevalent the condition is, how many more people, or even different sort of symptoms that do come out. I know with Alana, she's got a cortical vision impairment. It's not part of literature, yet in the community, there's about 5% of them do have cortical vision impairment. So it's clearly is significant, yet it's not been updated. So, you know, yeah, as clinicians, I think it's important not to rely on sort of the Unique if it's outdated and things like that. So
yeah, thank you. And I think Pauline has just shared a link for Medics for Rare Diseases, which is an excellent charity sort of looking at trying to increase awareness of rare conditions. And Pauline, I think you also mentioned going to some conferences with a support group.
Yeah, thank you. I was going to point that out. There are actually patient advisory groups which have a lot of so that sort of Tony's just posted up about third sector charities. So patient charities or patient support groups are a really good source of information, and as Anna's found, sometimes you find them on Facebook because that's where people are, but some of the like, as I say, mine's a more common rare disease, so there are actually sort of patient charities that I think were started in 1973. And they have a lot of information, and they can help people to find other families that are going through the same thing. And sometimes that just finding someone else, they might not have answers, but knowing that you're experiencing the same thing can be really helpful. And this is the thing, the spectrum of rare disease is so wide. Mine's a very common rare disease, and then you get ones where there's one family in the country dealing with it. But as medics, there are sort of rare disease organisations out there that can help point you towards other sources of information.
Thank you, Pauline. And Helen, I think you've actually started a charity yourself, haven't you, to help others?
Yes, yeah, I have. Yeah. And we're very patient focused. So we run a coaching model. We don't think it's within our gift to tell people what to do, but we ask powerful questions of our community. And it was interesting looking, hearing Cara talk about the conversation she's had where, well, why did you have a section? You should always avoid why questions, because it puts people on the defensive. You'd say things like, well, what was your thinking behind that? So try and use what questions rather than why in your communications.
Thank you. Well, that's really helpful advice there. Isabel, I don't know, it's 2 o'clock now. I don't know if you wanted to close. I can't see any other questions in the chat.
Yes. So don't have any more questions. Sorry, honey.
Well, there's one question that's just popped up, I think. So.
Can't see the chat, I'm afraid. So
I think we've got one question that's popped up from Annabelle here.
Did you want to share it, Anna?
Yeah, so I have a question regarding your shared experiences of not being listened to or brushed off by various professionals. Clearly, there needs to be a push to listen to patients and family members who have lived experience of what is going on, and more education around genetics and how to recognise these things. Although this should absolutely not be on the shoulders of patients, is there a route for you to feedback through patient advocates to those professionals who missed critical things in your journey?
I was going to suggest that the patient participation panel, there's Helen here, that represents Genomics England, would be a good starting point. There's about 25 of us on that panel.
Good suggestion, thank you. I don't know if I can ask a final question, I don't know. So I was pleased to be able to help as a PPIE facilitator lead for the needed study pilot. And one of the concerns I have in trying to work out how to do the PPIE is to support those that were joining and participating and helping us, because I wanted to create a supportive environment. But would there be any supportive environment, knowing that there's concern of kind of emotional entwinement with the experience and knowledge sharing from those helping us? Is there any particular advice or any from those that have been involved with patient involvement groups, anything that you would offer to help those that are facilitating, to support those that they're reaching out to? Long-winded questions. Just wondered if there was a way to make it as comfortable and as supportive environment as possible, knowing it's a difficult process to go through.
I think where, I don't know if there is a right sort of play, but in person I think is always helpful. As I say, delivering, I think Anna has also, she said about having that like a bombshell over the phone. It's ideally you do need to get face to face, and I know some appointments can be done over the phone and sometimes easier over the phone, but when it's like big news, it needs to be face to face. It needs to have time to have those conversations, and potentially is there any way of expanding sort of, because I know like with the genetic counselling stuff, it's more about like on the genetic side, but is there more of a sort of the traditional word of counsellor being there? Don't think there really is that much sort of support, that emotional support. It's just, as I say, NHS is stretched, so to get that kind of where can you go to sort of sob and have a cry and have some tissues and have someone listen without paying private for that. So it's hard. It's I don't think there is a right way or a wrong way to be able to share difficult conversations. It's hard.
I wonder if, because I've got a bit of a unique, different experience in that I was born with the rare disease, so I haven't been the parent sitting there. But I guess one of my questions for sort of Cara or Anna or Helen would be, would a sort of accelerated follow-up have helped? So you have the initial conversation, and then once you leave that appointment, you then know that you've got something in the diary in the next couple of days, or you've at least got phone numbers or emails or something someone you can call when you start having more questions, because obviously you're not going to have all your questions in the first appointment, but if you've got a way of finding out more from a thank you, patient initiated follow-up, but also just something in the diary to make sure that you haven't left this appointment with no other way of asking questions.
That would be as ideal, really. I think it's like in one of my points I said about like having contact numbers, having the information of who can you go to ask questions. But if you had it as an actual appointment for a few days later, that'd be brilliant.
Thank you so much for all your thoughts. That's really helpful. So I don't think I can see any more questions in the chat. I don't know, Emma, can you see any?
No, there's
I can
no.
see lots of thank yous for all sharing your experience, though. So thank you again, and I'll just hand back to Isabel.
Thank you so much. So yeah, just to reiterate, thank you to everybody who's joined, and thank you to all of our speakers, in particular for sharing their stories. I think it's really valuable to do things like this. We will be sharing all of this recording and the slides after. We'll also be sharing information on Central and South Genomics. On the website, you can see more about our PPI panel. You can read some of our patient stories, and obviously we do encourage you to sort of read all of the information that you can on genetic and genomic testing as healthcare professionals, particularly to make sure that we're kind of getting a good understanding of supporting patients through that process. We will hopefully be sort of expanding this into a sort of creation of more materials, and so to that end we will be sharing a survey after. It will probably be next week that we get this recording and all the materials out to you guys, but we will be sharing a sort of survey for any of your views on how you would like those materials to be sent through, and what you would like them to look like. We want to sort of co-create those with people on our PPI panel and other sort of patients and advocates. So we're 7 minutes over, so I will say thank you again to everybody and wrap up this call now. I hope you have a wonderful rest of the day and a great weekend. Thanks, everyone.